Hi, my name is Dana from the University of Queensland, and I'm going to talk today about using brain imaging and machine learning to explore potential biomarkers for dementia and Parkinson's disease. 80% of people living with Parkinson's disease will at some point develop dementia. However, we're not sure why some seem to decline in cognition faster than others. One theory is the dual syndrome hypothesis, which proposes that there are two cognitive syndromes in Parkinson's disease related to dysfunction of the frontal or posterior regions of the brain. The frontal syndrome presents with deficits in executive function and attention, whereas the posterior syndrome presents with deficits in memory and visuospatial function. And it's the posterior syndrome that's predicted to decline more rapidly than the frontal syndrome in terms of cognitive ability. To identify these syndromes in our sample of 85 Parkinson's patients and compare their brain activity, we used a machine learning method called k-means clustering. We entered everyone's data from 10 frontal and posterior variables into the clustering algorithm, which then told us whether each patient was either frontally impaired, posterior impaired, not impaired at all, or globally impaired on almost everything. This allowed us to avoid using arbitrary cutoffs for cognitive measures to identify the subtypes and derive completely data-driven groups. Given that there was existing brain imaging data for a subset of these patients, we then explored differences between the posterior group, remember that's the at-risk group at risk of imminent dementia, and the cognitively intact group to see if we could identify any neuroimaging biomarkers for the at-risk group. Through analyzing the resting state functional magnetic resonance imaging data, which just looks at brain activity at rest, we found that the posterior cortical group demonstrated weaker connectivity between the right hippocampus and the right anterior temporal fusiform cortex at rest compared to the cognitively intact group. It's important to acknowledge the limited sample size of the data set and mention that we were unable to compare the posterior group to the frontal syndrome due to limited numbers in the frontal group. However, this finding is nonetheless interesting because if you look at the role of the fusiform cortex, it appears to be involved in not only episodic memory, but also semantic and visuospatial memory. And if we look distinctly at the measures that the posterior cortical syndrome were impaired in, we can see marked differences in each of these three memory processes. Like any interesting finding, the results leave more questions than they have answered. For example, what's the role of the fusiform gyrus and its connectivity to the hippocampus in the development of dementia and Parkinson's? Well, moving forward, we're planning to look further into this potential biomarker using larger longitudinal data sets with the hopes of developing a powerful and clinically useful tool for identifying dementia risk in Parkinson's disease. Thanks for your time, and please get in touch if you have any questions.